Good morning, everyone. My name is Dave Stevens. I'm the Vice Dean in the Faculty of Science and a professor in the Department of Mathematics and Statistics. On behalf of the Faculty of Science, I want to welcome you to the 30th edition of Soup and Science. Soup and Science is the faculty's showcase for research of the professors and students in the faculty. And over the next hour, we'll see examples of the quality and breadth of the research that's going on. But first, it's important that we acknowledge that McGill University is on land which has long served as a site of meeting and exchange amongst indigenous peoples, including the Haudenosaunee and Anishinaabe nations. We acknowledge and thank the diverse indigenous peoples whose footsteps have marked this territory on which the peoples of the world now gather. This morning, we have four great speakers who will be presenting their research. We've got Professor Peter Douglas from the Department of Earth and Planetary Sciences, Professor Hans Larsson, who's the director of the Redpath Museum, Professor Catherine Potvin, who's a professor in the Department of Biology, and Émilie Saucier, who's worked on a project in the Department of Earth and Planetary Sciences. So once the presentations are finished, then they'll run for about 5 minutes each. You'll receive a message from the host, Preeti, who will ask you to join a breakout room. In the breakout room, there'll be smaller groups and you'll be able to interact with the speakers, ask questions in the chat box, and so on. The presenters will rotate through the rooms every 10 minutes. And then once the breakout rooms are finished, we'll all come back together again for a final sum up. And there'll be opportunity to again fill in a questionnaire at the end. So we'll begin with the first speaker, and the first speaker is Professor Peter Douglas, Professor in Department of Earth and Planetary Sciences. Thank you, Peter.
Okay, thanks, Dave. Let me make sure I can go through. So I'll be talking today about my research on using rare isotopes to understand climate change. Thanks, everyone, for being here. So what is an isotope? So isotope is an atom that contains different numbers of neutrons with the same number of protons and electrons. So it behaves the same in terms of its charge, but it has a different mass. Just as a quick side note, some of the earliest research on the existence of neutrons and isotopes was done at McGill by Nobel laureate Ernest Rutherford. So we've got a long pedigree with this. And I'll be talking today about all about the isotopes of carbon. So most of the carbon in our bodies and on the planet is carbon-12. It has 6 protons, 6 neutrons. But a very small fraction, about 1%, has an extra neutron. So this is carbon-13. And this means that it behaves a little bit differently in chemical reactions, and it's distributed a bit differently around the planet. And then there's another very small portion of carbon that has an additional neutron, carbon-14. And we all know of this as radiocarbon. But because it has so many neutrons, it's actually not stable. It breaks down over time. It has a half-life of about 6,000 years. And this is very useful because it serves essentially as a clock for how old a pool or a reservoir of carbon is. So what does this have to do with climate change? So I use isotope measurements to study climate change in a wide variety of contexts, but I'll be talking today about my work on greenhouse gases. I'm guessing we're mostly all familiar with greenhouse gases, and the two most important ones are carbon dioxide, which we're probably most familiar with. There's quite a lot of it in the atmosphere. It's going up. It's the prime cause of climate change. And then there's also methane. And methane, there's a lot less of it. It's also going up in the atmosphere. But despite its relatively small concentration, it's responsible for about 25% of global warming. So it's quite important. Now we're coming up on Halloween, and these are the two bad guys in our talk. So I put some funny faces on them. But the scarier face on methane just is there to indicate that on a molecular basis has a much stronger effect on warming. So it has about 35 times the warming effect of carbon dioxide. So again, it's quite important to understand where it's coming from and how it's getting into the atmosphere. So what do isotopes have to do with this? So when we look at a molecule of CO2 or methane in the atmosphere, we can't really tell where it's come from just based on its chemical structure. But if we delve a little deeper and look at the isotopes, either of carbon or of the other elements in the molecule, this can tell us more about the source, so where these molecules are coming from, where these gases are coming from, and also about their sinks, where they go when they exit the atmosphere, and that's also quite important. So we start by talking about methane, and methane's quite interesting because, or it's mysterious, I guess. It's done some interesting things in the atmosphere over the past few decades. So I don't know if you can see my mouse, but beginning in the 1990s, methane actually stabilized in the atmosphere, and it was stable for about 10 years, so it stopped increasing. And then about 10 years later, it started going up again. And scientists have studied this quite a lot, but we still don't have a great answer as to why it stabilized and then began going up again. And isotope measurements have a lot of potential to help us understand this. I've been focusing a lot on radiocarbon measurements of methane, and what's very powerful about this is that when you have fossil fuel methane, it's too old to have any radiocarbon. It's all already decayed away. So when you have fossil emissions of methane, they have no radiocarbon. But if you have emissions of methane from wetlands, which are another large source of methane, they have a lot of radiocarbon. Now, I also have this box here for permafrost, and permafrost has been sitting around for thousands of years. So it has less radiocarbon than a modern wetland, but it has more than fossil fuels. And in Canada, we have kind of two key sources of methane that are of concern that I've been focusing on. So one is extraction of fossil fuels, especially in Alberta, which produce quite a lot of methane. And then also the thawing of permafrost. So Canada contains a wide expanse of permafrost. When this warms up, it will release greenhouse gases, including methane, into the atmosphere. So we've been doing research in northern Quebec, in Cree territorial lands near Wapmagoosty-Kuujjuarapik, and there is widespread thawing of permafrost, and this has led to the formation of lakes that produce lots of methane going into the atmosphere. And the question is, how much does the actual permafrost carbon contribute to these methane emissions? And what we found is actually most of this carbon is what we would call young carbon. So it's actually not coming primarily from permafrost. Instead, it's coming from all this nice green vegetation that we see. And so at least in this one specific case, we don't have this big permafrost feedback, and actually most of the methane is coming from the plants in the area. Now, we're also working in the oil sands of Alberta, and this is a very interesting situation because we know the oil sands produce a lot of methane, but they're also surrounded on all sides by bogs and other wetlands, and so they could also be producing a lot of the methane that we see in the atmosphere there. And so I've been working with Environment and Climate Change Canada to do measurements of radiocarbon in the atmosphere, in methane, to parse out exactly how much of the methane that's coming into the atmosphere there is coming from the oil sands, which will help us ultimately to better control this emission source. We don't have the data yet, so I don't have an answer to my question, though. So I want to just finish up, and I know I'm probably running a bit low on time, with another side of my research, which is looking more at sinks of greenhouse gases, and in this case, carbon dioxide. So we know that plants, and trees in particular, are an important sink of CO2. Much of the CO2 that we emit into the atmosphere does not stay in the atmosphere, but goes into this land sink, which is primarily plants. But what we don't have a great sense of is how this is going to change in the future when we have very high CO2 concentrations in the atmosphere. One of my PhD students has come up with a really exciting natural experiment to look at volcanoes as an analog for an environment with very high CO2, because volcanoes produce a lot of CO2, and they also often have forests growing on their sides. And so what's also very useful about volcanoes is the CO2 they produce has a very distinctive isotopic composition. It contains a lot of 13C, or carbon-13. And so on this plot, you can see we're up here with these values that are indicating lots of carbon-13. Normal forest CO2 is somewhere down here. There's much less carbon-13. And so the goal of this project is to look at tree rings in trees from these volcanic environments to see how much of this volcanic CO2 the plants are taking up. And hopefully this will help us to understand what might happen in the future when the whole planet has much higher CO2 concentrations. So I'll leave it at that. Feel free to get in touch if you're interested in this research. And here's some photos of my grad students off in the field doing fun research.
Great. Thank you very much, Peter. Together, we'll move on to the third speaker then. So the third speaker is Professor Catherine Potvin from the Department of Biology. Thank you, Catherine.
So good morning. Bonjour, Kwe. It's a pleasure to be here, albeit virtually. Thank you for the invitation and the organizers. So I'm a biologist, and it's sort of nice to speak right after Peter, because my talk has some bearing with climate change. I'm unable to change the slides, so maybe somebody, okay, now I see the second one coming in. So I'm a professor in biology. I'm a botanist by training, and I hold the Canada Research Chair on Climate Change Mitigation and Tropical Forests. So there goes, I'm not sure I would say two of my passion, but certainly a passion for forests and a very big concern for climate change. And I thought I would bring you to the very most recent work that we're doing in my lab. It's really trying to assess how big is a tree. So you see a picture of a good friend of mine. It's a tree that fell over a river, and it sparked an entire different piece of research for me 2 years ago. Sorry, again, I'm having trouble changing slides. So I'm hoping to go to the second slide. Peter was talking to us about the increment of methane and carbon dioxide in the atmosphere. So most of us, we know this, this graph, which shows, I hope you see my mouse, the increase of carbon dioxide since the industrial era. But superimposed on this slide is a kind of a wave in red. And I call that the breathing of the planet, because people don't tend to know this so much, which is that forests and land all around the world are having a seasonal impact on carbon dioxide. So if you were to follow these red oscillation, when the carbon dioxide concentration is very high, it corresponds to the winter of the northern hemisphere, where trees are largely leafless. And during summer, we see a drawdown. So we see carbon dioxide being actually captured by trees, and it has a global signature. So trees act on the atmosphere in this way. They take out carbon dioxide from the atmosphere, and they store that in their trunks. So forests are a major reservoir of carbon, and hence an important way of regulating climate. A couple of years ago, a paper was published talking of natural climate solution, and it made forward the idea. I'm sorry, I don't know why the slide is changing. So I'm still talking of the previous slide. I'm not sure, but you saw that. So there, okay, I'm not sure why my presentation is just going on alone. So natural climate solution is the idea that the only technology we know so far is photosynthesis, if we're talking about cleaning the atmosphere. So it's been on the eyes of policymakers to use carbon sequestration through either planting trees or preserving forests to act on climate change. If we want to act on climate change efficiently, then we really need to be able to quantify how much carbon there are in trees and in forests. And that is much more difficult than one would think. I mean, if we could go and cut forests and put trees on a scale, we would actually be able to know how much carbon there's in a forest, because half of the weight of a tree is carbon. But if we want to conserve forests, and if we want to, to plant trees to sequester carbon, of course, we cannot go and harvest them. So there's currently a range of different technologies that are available to try to get at this. And they go from very, quote, simple technologically, but challenging to implement. You see on the right side of this slide, two men working with me trying to measure the diameter of a tree. So that's hard physical work. And here is a picture of another way in which you can measure carbon, which is using a terrestrial LiDAR scanner. So this uses laser beam to bounce on objects in a forest. And through that, we can reconstruct a three-dimensional, size of trees and quite precisely measure those trees. I'm mostly working, doing field work in Panama. This is a small Central American country. It is bordering with Colombia on the east and Costa Rica on the west. This is a map that we did of the carbon stocks of Panama using an airborne LiDAR. So a plane flying this same technology that you see below from the air. And you see that we have at the border of Colombia area of very, very dense forest carbon. So in the past 2 years, my students and myself, we've been trying to provide some better understanding of how much carbon really are there in these forests. I'm trying to change slides, so we'll see if that works. I'm sorry for the technical problems here. Okay, yes, so I think the next, the next slide is there. Hopefully it's gonna stay there. So as we were going to these very far away forests, we came across this tree, which I mentioned earlier, had fallen over a river. And what is so special about this tree is try to measure the height of a tree. The height of a tree is an important element of assessing its biomass. On the right side of the screen, you see here a terrestrial laser scanner of a tree in this forest. And this is the people that were measuring them. So you can see at scale the size of the tree and the size of the person measuring it. So in a forest, it's almost impossible to see the top of a tree and estimate its height. So this particular tree had the advantage of being fully horizontal. So we were able to measure it. And it measured 71 meters in height, which is amazing because it's about twice the height of the reported forest of Panama. So, you know, it's not just a small mistake or a small error, it's huge. Here you see some of us in the field team, my field team, standing on top of the tree. We were 22 on this picture, and we could have put more people. But what is more special about this particular tree and many other trees in this forest is now that we were able to measure it well, we know it contained 50 milligrams of carbon. That's a single tree. Well, in Quebec, in the temperate forest, the estimate of the government of Quebec is that there is 40 tons of carbon, 40 megagrams of carbon in 1 hectare of forest. Okay, so 1 hectare is 100 meters by 100 meters. So one tree in Panama is more than 1 hectare of forest in Quebec, which explains why I'm really working there. Trying to move on. The question, if we want to really see in this forest, how much carbon is there at the landscape level, we have to scale up. So we have to measure trees, and then we need to know how many trees are there on the landscape. This is a very useful thing to have to do during the COVID, since we cannot go to the field these days. So we've been using high resolution satellite imagery to identify big trees. If you look at the panel with 6 pictures, look at the top figure where my mouse is, you can see the fallen tree that was still alive over the river. It means we can see the tree, an individual tree, from the sky. And here you see, or maybe it's harder, you actually see the trunk going across the river. So we know that this tree fell between October and December 2018, exactly. So having proven that we could see individual tree from the sky, we began a very tedious analysis of a series of high resolution imagery of the forest. Sorry, my slide moved again. So we began an analysis of a series of imagery on the forest. And look at this spot here. You can see it on the first corner to the left below. And as you go, you can clearly identify this tree in many images. So we looked at the landscape level at a region of about 25 kilometers by 20 kilometers. And we identified over 200 trees that we believe are as big as the fallen tree, and actually went in the field to verify their size. So we are getting now the information that is proving that this forest is absolutely exceptional in terms of the carbon it stores. We call that a refugia, a place where trees and forests grow much bigger than is believed to exist. And this is a novelty now, you know, that we have much better, remote sensing tools. Everybody working in tropical forests is realizing that there are pockets of abnormally big forests that have extremely big stores of carbon or reservoirs of carbon, and that are very important in terms of maintaining the balance of carbon at the landscape level. So the next step in this research, and my last slide, is the fact that we're working with the villages. So this is in an indigenous territory. So we are working with the people of the village to implement a biodiversity survey and carbon inventory at the landscape level. We're using a mixture of instruments. So the jaguar you see on the bottom of my slide was taken by a camera trap. Very excitingly, we're pioneering a technology that is using soundscapes. So we put recorders for sound in the forest, and we're recording sounds over continuous periods of months to develop metrics of diversity from that. And it's all the people in the villages that are operating the instruments and the monitoring. Our goal is to establish an old growth forest observatory and find ways to bring some income to the community and support them to protect these exceptional forests. So thank you very much.
Thank you very much, Professor Potvin. So our third speaker will be Émilie Saucier, who's worked on a project in the Department of Earth and Planetary Sciences also. Émilie?
Hi. So yeah, I'm in my third and last year of honors major in geology, and I wanted to share with you today all the opportunities that I've had the chance of experiencing in my department, whether it is field trip, research, or just classes that are really awesome, and how that spans all my 3 years at McGill. So we first started my first semester with a class called Geology in the Field, where we go and do some little field trips all around Montreal to see mostly road outcrops, where the geology is well exposed. So that was our first introduction to the geology in the field. And then at the end of our first year, we go to California and Nevada, where we can experience a geology that isn't covered with vegetation like it is in Quebec. And that was also quite spectacular. So we have a bunch of these classes that brings us to the field so we can experience geology there. We have also a bunch of field opportunities that are optional trips. The most popular one is called the Willie trip, which is organized by one of our professor, Willie, who comes with us to contribute choosing. So the undergrad students basically organize a geology-oriented trips, and then he comes with us during 2 weeks during March break, and then we explore the geology of a country with the pace we want and the way we wanted to do it. So that's a great way to learn. And this year we went to Morocco. And then the previous year, we had gone to Kenya and Tanzania, which was also quite a spectacular trip since we were right in the Rift Valley. So we had a lot of cool geological occurrences. There was this volcano here in particular, Old Anyolanga, which is a carbonatite volcano, which was pretty cool to see. During this trip, we came across a weird geological occurrences called an obsidian dike, and our professor suggested that one of us bring back some rock sample of this to Montreal to do a research project on it. I was the lucky one who got to do it, so we brought the hammer out, took some rock samples, brought them back to Montreal. So at the time, I was just in my second semester of university, and I already have a research project lined up just because I decided to go on this optional, very affordable trip with the department. So there was that. And then that was my first research experience. The second one is what I did this summer in the Sura with the McGill Volcanology Research Group, where I did two main projects. The first one about volcanic gases and the standards needed to measure their content in volcanic rocks. And then the second project I did was about some vesicularity in pumice clasts from samples coming from Iceland. And that was a great experience, and it actually inspired the honors research thesis that I'm doing right now. So I now have my own sample from Iceland that I'm going to do all the process from beginning to end of analyzing, once again with the vesicles in it, which are the little holes that are left by the rocks that crystallize around the bubble in a volcanic rock. So that's basically how I've been spanning my years at McGill with research and fieldwork and opportunities that are amazing. Yeah.
Thank you very much, Émilie. So we'll now go back to Hans with fingers crossed for the final presentation today. Back over to you, Hans.
Great, thank you very much. Apologies for the technical difficulties. When there's no internet, there's no internet. So I'm calling through cell right now. So if Preeti, if you can advance the slides for me, because I have no control on that. So first slide, please. So welcome. I'm the as Dave was saying, I'm director of the Redpath Museum, and I'm also a vertebrate paleontologist. And so my background is fossils, geology, biology, and as you'll see in a bit, also experimental embryology. And so, kind of in a nutshell, this whole presentation is really what can dinosaurs do for you? Next slide, please. And as I mentioned, I live in the Redpath Museum. I direct the Redpath Museum, and this is something that some of you who haven't really had a chance to be on campus will certainly experience once we come out of red and orange zones. Redpath Museum is one of the oldest buildings in the museum. It's the oldest natural history museum in Canada, and we have a very strong public program unit where we work with about 100,000 visitors per year. And so this is both in-house, out-house, like in terms of outreach to schools. And so we have a very active public programming unit where we also involve lots and lots of undergraduates and graduates, students in the building. Next slide, please. And it's also quite central to McGill. So this was literally a year ago. So quite different today, where campus is all about abandoned. But really, the Redpath Museum is so central to many things functioning in and around McGill, both from Faculty of Arts, Faculty of Science, Faculty of Engineering, Faculty of Medicine, and even music. Next slide, please. And so for the next couple minutes, I just want to do a brief sort of synopsis on some of the research that's gone on or published in the lab in the last 7 or 8 months. And so the lab is relatively broad. So we work on developmental biology, evolutionary theory, geology, paleontology, and so forth. But I just want to start off, next slide, in the field. And my lab and myself are field nuts. We worked on every field station that McGill has. We're helping to create more field stations for other museums and explore anywhere we can around the world. This summer, every summer, typically I run, except for this summer, I run a field course in Western Canada where I drag a couple dozen undergraduates and some graduates through the Badlands of Saskatchewan and Alberta, where we go and break rocks, discover geology, and collect dinosaurs. Next slide. And from that, gain, I think, a really good appreciation from the foundations of science. So going out in the field, camping by rivers, living in a tent for 3 weeks, eating nothing but fire-cooked food, and learning what geology and fossils can tell us about the world around us. Next slide. And so, while dinosaurs seem like a bit of a weird research to be happening inside of a cutting-edge university such as McGill, they do have a role to play. And I'm not going to shy away from the fact that 3 out of 5 kids, children, experiment with dinosaurs in terms of interest in STEM, so science, technology, engineering, medicine. And so I really hold true that anything published with dinosaurs, although it has very little effect to the real world, actually can encourage kids to come into STEM education. And so here's a paper we just published. We're looking at, while it seems like it's a bit esoteric, how fast could a T-Rex run? What we're doing with this is actually modeling how much energy does it take for a T-Rex to run? How much energy does it take for a flock of these animals to survive? And so now we're going through some of the geological formations that we've been working in and trying to estimate what is the actual energy flow through these ecosystems, in these paleo ecosystems, just before the mass extinction 66 million years ago. Next slide. And of course, it plays well into media. And so this has millions and millions of people viewing this and documentaries made on this from this one paper. So a way to attract lots of kids into the field of science. Next, please. And it doesn't stop. So we do research in as many field sites as we can. So here's a site that we've been working a lot on in Colombia. And we just described this enormous, close to 10 meters long crocodile. New species, brand new animal for Colombia. Next slide. And it gets bigger. So this is just the head of an animal that we ran across in a sort of like a private school / church / museum in this faraway town in Colombia just before the lockdown. And so this is another animal that we're working on. To give you a size for the animal, that's just the head. So the head is pushing 8 feet long, easily one of the most terrifying animals in the water at the time. Next slide. And they're everywhere. So everywhere we go, we're just finding new species. This is another animal from Colombia with one of my Colombian graduate students. Next slide. And to make some use or lemonade out of lemons, what we've been doing for the last several months is reconstructing one of these animals. Here's a species from Saskatchewan, and it's a full skeleton. It's about 14 meters long. But we've gone through and surface imaged the entire skeleton, and now reconstructing it into like a real sort of deformed position. And now we're printing these bone by bone. So I have a printer in my basement right now, where we're turning off, trying to print the skeleton. And when it's done, hopefully by around next summer, we'll be able to start mounting this in the Redpath Museum. So a 14-meter long Canadian plesiosaur. Next slide. And to add to the mix, because of our work with comparative anatomy, macroevolution, I have a grad student from Mexico where we've been looking at crocodile population genomics. So we've sampled several hundred crocodiles, and this has been an amazing field experience. I've always wanted to wrestle crocodiles before, not just fossil crocs. And so this was a great experience to learn how to capture and do good genomics work. So we're using the Genome Quebec facilities to actually sequence the full genome of a crocodile for the first time. And also this plays directly into the conservation status of these animals in various populations across New Mexico. Next slide. My lab also runs a radiology facility. It's one of the only nano-focused micro-CTs in the world. And so with that, we've been doing a high-level comparative anatomy. And so here's just a snapshot on some of the anatomy that we're working on. A lot of it is bird anatomy, some reptile anatomy. And the reason that we're doing this is that by looking at bird embryology and bird anatomy, we're actually describing dinosaur embryology and dinosaur anatomy. Next slide. And why this is important, not just for the sake of comparative biology, but also we've been teaming up with colleagues in chemical engineering to take our imaging toys and our love of comparative anatomy and comparative embryology and apply that to real world questions. So what happens when you ingest artificial astroturf or artificial athletic turf? And so many of you have played on this. Many of you have probably smelled it, and you see the black crushed up tire rubbers inside it. But what does it do to you? Not a lot of research out there, and certainly nothing that I would say is relevant to human physiology. So what we did was we took chicken eggs, fertilized eggs, and actually sort of exposed them to some of these microplastics. And athletic turf is actually considered a microplastic in the Canadian health definitions. And on the bottom is a normal growth series of embryos, and in blue is one of the embryos that sort of grew out of this artificial athletic turf. And you can see it doesn't look normal. In fact, the brains didn't even separate from the eyes on this embryo. And we found that about 50% of the embryos actually are lethally deformed growing on this athletic turf. So that was a fun paper and hopefully of relevance to how we go forward and treat a lot of these synthetic environments around us. Next slide, please.
Hans, you have about 30 seconds left.
Sure, I've only got 2 more slides. And the other sort of spin-off on this was that we started thinking about microplastics. And so we published a paper in December last year where we discovered that each cup of tea of an expensive kind of sort of silk tea bag actually produces billions of nano and microplastic particles. And this is orders of magnitude what we thought that humans were consuming annually. And so we're finding that we're compressing this into 1 cup of tea. And this also has, of course, health effects, because we found that Daphnia, this little freshwater crustacean, develops deformities, and it also has behavioral alterations as well. So this is just the beginning, tip of the iceberg on how we're now applying our imaging toys and tools to microplastics and environmental toxicology. Next slide. And finally, the teaching is a big deal for McGill and how we go forward. And because a lot of my teaching is either field-based or lab-based, how to bring that in the world today. Just before, on the bottom, you can see students that are dissecting an alligator, because this is how we do comparative anatomy, like getting elbow deep inside carcasses. So now, of course, we can't do that, although I'd love to ship alligators out to everybody and do it remotely. But so now we're surface scanning and imaging lots and lots of museum specimens, so that everyone can look at this material in almost as good resolution on their own screens at home, and do the same kind of lab exercises. So thank you.